I hope everyone's enjoying your dinner. Our next speakers are going to talk about a unique program for Schoolcraft College. It's the Undergraduate Biological Research Internship Program. Very unique because it actually gives our students the opportunity to get involved in biological research. So I'm going to welcome to the stage Dr. David O'Hagan, a biology instructor, and Mackenzie Hayes, a current student of Schoolcraft.
So if you don't mind, just take a peek at this video. This video was produced by Steve Maloney, and I want to thank him for doing that for us. And here we are, working in the lab, young minds inspired. And there's Roger Newton, who's going to be talking to us today about his experience. And there's our students for this year. And I just think that's a great video. So, thank you. So everybody's eaten and everybody's full. And what's good about this presentation is that all you have to do is look at pictures. It's and they're all pretty pictures. So what I've done is I've taken a half-hour presentation, I've squeezed it down into 5 minutes. So all right, there we go. So what I would really like you to do is to, enjoy the images. They're wonderful. And then we have Mackenzie Hayes that's going to present one of the projects that we're working on that actually worked, something that we never thought would happen. And she was instrumental last year in making that happen. So I wanted to make sure that she had an opportunity to explain what it is that she did. So let's just start by, I just want to explain a little bit of how the program started and why we started the program. So it's well known among educators across the nation that coupling practical application with traditional classroom tutoring is the most powerful way to get a deep, thorough understanding of the topic of interest. And so we knew that ahead of time. And so we tried to figure out ways in which to engage students in the classroom. And it's very difficult. You don't have any equipment around, and the equipment that you have is not what you would find in industry. It's not what you would find doing your PhD. It's not what you would find if you were doing the job that you were supposed to do when you graduate. So we do our best. And while we were, I was teaching a genetics class, I had 2 students that had a genetic disorder. One girl had very fair skin and red hair, and the other girl had a clotting factor mutation. And they came to me and they said, well, how do we cure this? I said, well, you can't. There is no cure. I said, well, if we wanted to get a cure, if we wanted to create a cure, how would that happen? And I said, well, you know what I want you to do? I want you to take the next 2 weeks and I want you to go out and look at the research and tell me what you think. Now, granted, these are freshman, sophomore students. This is their second biology class, basically. And they came back and it astounded me. It blew my mind. I couldn't believe what they were suggesting because I couldn't find any problem with it. I couldn't find out why it wouldn't work. And they said, well, can we test that? And I'm thinking, oh my God, I got to get out some stuff in my garage. And I'm thinking, wow, how is that going to work? And I thought, well, with a little bit of help, with a little bit of luck, and a little bit of money, we probably could make that work. So I want to point out this strategy slide. What we asked them to do was to, let's see if I can point this so everybody can see. So we asked them to identify a problem. And then after identifying a problem, I asked them to do some investigation of their own. And then after that, I said, use your classroom instruction to come up with a solution. You guys come up with the solution, and I'll help you make it work. Okay, So here's Nicole Driscoll and Kirsty Schofer, our two students in 2011 who started this program. I didn't start this program, they started this program. They pushed me to make this happen. So when we decided to move forward in these two areas, one was it's deep vein thrombosis. It's a disorder that can lead to pulmonary embolisms, strokes, heart attacks. It's very serious, and people with clotting factor mutations are susceptible to these diseases. And this is what Mackenzie's going to tell you about. So I'm not going to talk about that anymore. The other one was she wanted to tan. It wasn't anything more than that. She had skin, she wanted to go out, she wanted to tan. So that's a receptor, makes you tan. And unfortunately, her receptor was mutated and it hides away inside the cell. So we came up with a way to figure out how to kind of bring it to the surface of the cell, and we're still working on it. That one's a toughie. Anyways, to get this working, we've got our administration that without their help, without their blessing, there's no way that this could go forward. And I just want to recognize Robert Ledley, Rich Weinkoff, and especially Thea Greenshields. She is in the dean's office, and she is single-handedly doing all of our budgets and our making sure that the orders are in and paying the people that need to be paid. And I want to give her a special thanks. So we started off there. And then very shortly after, we realized this is a teaching moment. This is what we were looking for in the classroom. And so we started the Undergraduate Biological Research and Intern Program. And you probably all know it because you saw my application. So this application was joint funded by the foundation and the Michigan Life Sciences Innovation Center, which was Roger Newton. He was the one that gave us our space. And so this joint partnership, for the first time in Michigan, and to the best of my knowledge, nowhere in the United States does this exist, where you've got academia and companies working together to educate students and to prepare students for the next round, especially in biology, maybe in other areas, but not biology. Okay, so this is just the ad that came out at the launch of the program. And at that time, Spark, Ann Arbor Spark, was part of the Michigan Life Sciences Innovation Center. So let me just show you what this building looks like. Now all the pretty pictures. So here's this beautiful building. It's a state-of-the-art research facility right next door. It's on Beck and M14. I found it just by driving around because I just like looking at cool research buildings. And I don't know, it's one of those things. And so I drove and I looked at that, I said, wow. So I went in and I got Fred Earl. He was the manager of the space, identifying people that would fill that space. And he showed me a full kitchen, this incredible dining room, a workout gym, showers, lockers. I'm like, wow, if I would have done my PhD in a building like that, I might never have graduated. But he also showed me the most important thing, and that's the equipment. The institution, the Michigan Life Sciences Innovation Center, is truly a state-of-the-art area to do research from large companies, multi-million-dollar companies, and tens of thousands of dollars, early startup companies. So there are literally, there are so many companies that fill this innovation center. It gives our students a great breadth of different opportunities. And here's just an example of our students culturing human cells. This is a biosafety level 2 hood. You can't culture human cells without this hood because of viruses and things that would hurt you. And training on this hood is respected worldwide. Every biological researcher has to learn how to do this. And if you're good at it, you really get a feather in your cap. So here we are, we're culturing human cells. We're testing human cells with drugs that we designed, which was amazing. Here's where most of the hard work happens, and that's pipetting. And we go through lots of kits and lots of pipettes. And this is the expense that the grant that you supported us with gets used for. This is the largest of our expenses. And I just wanted to show you what a beautiful room that was. And so here they are using up your money. Okay. The other opportunity for the students is our internships. So they get the ability to do internships. Saeed Ahmed, he's not in our program anymore because he went to med school. But he had a bachelor's degree. He needed to get retooled. He needed to get retrained. And he came to me to learn some undergraduate biological research. No, he wanted to learn how to pipette and culture cells so he can go get a job. Well, anyway, so he ended up interning for a cancer diagnostics company called De Novo, where they look at leukemia using microfluidics. He learned a lot, and he worked there for a year. And then they started paying him, which was really good. I just want to call out special thanks to Arthur and Diane Rockall, because last year we were starving, we were dying, and we needed some extra funds. And they came in and they single-handedly lifted us out of the doldrums and allowed us to finish the work that Mackenzie's going to tell you about. All right. So I've explained how Roger came in and gave us all this wonderful space, but what I didn't tell you is the amount of equipment that he's given us over the years. Throughout the last 3 years, and I don't want to say this because I don't want to hurt anybody's feelings, but he's given us, from my point of view, almost $100,000 worth of equipment. And that microscope there, it has little lenses that are $10,000 a piece. They're really, really expensive. And that's just one piece. That's just one example. We just got almost $5,000 worth of glassware, and he was like, it was in a room. He just said, why don't you take this? It's amazing, and I just want to thank you, Roger. It's incredible. So before I hand this over to Lindsay, I just want to go through 2 other projects very quickly because they're half of our team works on them, and they're just so exciting. It's amazing what these kids can do, especially when you tell them or when you don't tell them what they can do. If they don't know they're not supposed to do something, they come up with the greatest ideas in the world. All right, so here we are a couple weeks ago, and we have a chemistry professor, Michael Waldyke, and Danielle Ramos, which is here. She's from Canton High School. She is giving us Canton High School students that are joining our program now to get them even earlier, even younger. So I wanted to let you know that they are participating with this. And this is our biofuels group. And so the biofuels group is taking algae and they're making gasoline. Isn't that incredible? Okay, they haven't done it yet. But if it started off with a group of people and a paper that they brought to me. They said, well, hey, you can take these gut bacteria, these E. coli cells, and you can make gasoline out of them. Why don't we just do something that uses photosynthesis and just cut out the sugar, cut out the middleman? And I said, okay. So what we didn't realize is how difficult it was going to be. I can't explain how difficult these molecular pathways are, but their brains are nimble and they can think that hard stuff. And so they did, and here's just an example of their overall pathway that they presented to Roy Curtis at the Arizona State University and Danny Ducat at Michigan State University, where they created a joint research collaboration using algae from Roy in Arizona and Danny's lab in Michigan State University to create biofuels. The biofuel on this over here, this is what Roy makes. He makes diesel fuel. We're going to modify his algae, and we're going to make them small and turn it into gasoline. It's a great fit between our biological and our chemistry departments, and it's hard. Sometimes it's difficult for our departments to work together, but this is a natural fit. This is a win-win situation for both of us. We also are now part of a National Science Foundation grant. We're getting money to help support our lab work at Michigan State, and we have one of our students there that's working at Michigan State as he transferred. He transferred from our program into Michigan State, and now he's in the lab, and he's still part of our group. We have collaborators across the world. We've got Rick Sturm in the University of Queensland, Australia. We've got David Ginsberg at the University of Michigan, Roy at Arizona State University, Michigan State, Danny Ducat, and actually this is not the complete list. So last one before we hand it off to Mackenzie. They want to cure cancer. Who doesn't, right? Tall order. But don't tell them what they can't do, and they'll do it, right? So here's their plan. Chemotherapy, it's very toxic. It's very painful. We go in with chemicals that are so toxic that it kills all cells, any cell that's dividing. Very painful for skin, hair, intestines, stomach. Very, and not always successful. A lot of cells are resistant to chemotherapy. So there is this new strategy where we can target genetic elements to specific tissues by using regulatory elements in front of genes that are normally expressed in those, like prostate-specific antigen. We know that that's a marker for prostate cancer. Well, it has a gene. It has a regulatory region. They want to take this regulatory region and stick it in front of something that I created while I was at the Karmanos Cancer Institute, and it's a modified enzyme normally used to chop up DNA while you're eating your food, but I sent it into the nucleus to destroy the DNA because no cell lives without DNA. And what it did was induce something called apoptosis or apoptosis. And this is programmed cell death. It's the same kind of cell death that happens between your fingers during embryogenesis to get rid of the tissue between your fingers. It's something your body can tolerate. Your immune system doesn't respond to it. And it's a softer, gentler way to kill cells. And if we can get that gene to target to the prostate, breast cancer, ovarian cancer, childhood leukemias, watch out, because that's what they want to do. All right, so here's the last group. This is our clotting factors group, and I'm going to pass this off to Mackenzie. She wanted to make sure that I mentioned that she is the project manager of the Factor V team. She is the president of the Phi Theta Kappa Honors Society. She's an honors scholar and a writing fellow, and she's a busy girl. Okay.
Thank you, Dr. O'Hagan. Good evening, everyone. I hope you've all enjoyed your meal. I'm really excited to be up here today, and I just love to extend my deepest thanks for everything that you've done for myself and my team. It's my distinct pleasure to stand before you today as a testament to how much, like I said, the Schoolcraft Foundation has done for me. As Dr. O'Hagan mentioned, I am the project manager of the Factor V team, and we're working on the project quieting the mutant Factor V gene. And I would like to tell you a little bit about what we're doing with Factor V. Factor V is actually one of the many factors that controls the clotting of the blood. So, however, there's an issue. Sometimes the blood will clot too much. And in cases with people with deep vein thrombosis, or DVT, or pulmonary embolism, such as Dr. O'Hagan mentioned, the blood clots too much, and this leads for some serious complications, and life-threatening ones as well. So it has made it a prime target for our biological research. 30% of patients with DVT, or pulmonary embolism, PE, have a mutation on the Factor V gene, which leads to this overclotting. This mutation is called the Factor V Leiden mutation, or FVL for short. This is the issue my team and I are trying to eliminate. Our goal with this project is to use siRNA, which is short for short interfering RNA, to target FVL and destroy the FVL protein. This will shut that mutant gene completely off and allow only the wild type, the Factor V gene, which is the good one, to be expressed. Thus, clotting will return to normal. When I became involved in undergraduate research with Dr. O'Hagan, I had never had a biology course before, a college-level biology course. And so needless to say, I was a bit overwhelmed. All of the technology and the lab procedures, they were all so new to me, and I had to work super hard to learn what it took to help my team reach a satisfying goal last spring. last spring, yes, we actually saw our experiment work. It was quite fascinating. We successfully managed to knock out that mutant gene using our siRNA and allow the good gene, the factor V, to be expressed. And so here you see a very simplified schematic of one of our experiments, the experiment that produced such amazing results. See this dark spot here, and let's see if I can get the mouse, there we go. This dark spot right here, there's no band. Why is that? It's because the FVL gene isn't there. The siRNA, which you see here, completely wiped it out. This is a really good thing. But if you look right next to it, bright band, which is good. That means the factor V version, the wild type, is still being expressed. So this is a test tube scenario, but it basically says that blood clotting resumes. So this was very, very exciting. When this happened, it was just like, wow, we could not believe what we were seeing. So working with Factor V and Dr. O'Hagan, it has really been very life-changing for me. And this accomplishment and all of this hard work, going through the disappointments and the frustrations, and to see this, and it was phenomenal. And I couldn't thank my team more than I possibly could imagine. They were amazing, and this year's team is just as amazing, and I'm excited to keep working with them. There is something that my chemistry professor, Dr. Waldyke, does like to call the cloud. It's the cloud of confusion, frustration, and dead ends, where you grow as a person through disappointing circumstances and failures. It sounds like a lot of fun, doesn't it? Well, needless to say, I have been in that cloud for numerous occasions over the course of this project, and I have learned a lot of lessons that I have been able to draw on in my many areas of my life, none the least of which is tenacity. I'm so glad I have stuck with Factor V and am continuing to work on it with, like I said, such a talented team of researchers that I have now. My experience with my project has also aided me as I continue working for my associate in science. This opportunity helped me to narrow down my major from biology to molecular biology, and I've established several important connections in my career. Factor V has opened many doors for me, including the exciting opportunity that I had last summer to conduct paid undergraduate research at the University